Alrighty, so it's 5:15 and we are late. Sorry about that. We had a lot of issues with Zoom connecting to YouTube. And that means we're going to do the stream here on Zoom. I'm glad so many of you found your way here, and hopefully we didn't lose too many. I'll ask all of you to mute yourself if you're not already muted, just to make sure. And I'll just do a brief introduction about both this epic series of talks and also about Rasmus Nielsen. My name is Anders Albrechtsen, and I'm part of what's called EPIC, which is Evolution and Population Genetics in Copenhagen. And this is our summer series. So briefly, this EPIC is a network for population genetics in Copenhagen. Mostly we have physical seminars, which is very popular because mainly of free pizza. If you would like to hear more about this EPIC network or join the meetings, then you can join the mailing list at popgen.dk/joinepic. And the people here organizing these seminars are mainly from the Department of Biology and the Globe Institute at the University of Copenhagen. And here is shown all the pictures of the organizers right now. And we'll take turns organizing the talks and the introductions. So today we'll have Rasmus Nielsen talking about monogenic and polygenic selection in the human genome, but we also have a lot of other really cool talks. They're going to be at different times, so early in the day at 2 o'clock Central European time. So next time we'll have Benjamin Peter, followed by Matteo Fumagalli, and then Tyler Linderoth, Lucy Van Dop, and Anna Safo. And I hope you'll enjoy all of this series. If the YouTube link had worked, then you could find it again at popgen.dk/YouTube, and you can see the whole program at popgen.dk/epic. So, but today's talk is from Rasmus Nielsen. So he's a professor at integrative biology at UC Berkeley, and I'm very happy to be able to introduce him. Rasmus has played a very important part in building up the population genetic community here in Copenhagen, which has grown dramatically the last 15 years. And most of you listening now will probably know some of his work, especially in molecular evolution and population genetics, which is his highly used methods for both detecting natural selection and analysis of NGS data and much more. He's also produced some of the really cool works about understanding both animal and human evolutions, and most of you probably know about Tibetan adaptation to high altitude. He doesn't just produce great science, but has also played, and perhaps even more important role in producing great scientists throughout the world, as his role as a mentor and supervisor. Myself, have been lucky enough to be one of his students more than 10 years ago, where he introduced me to the awesome world of population genetics. So many of us here are here today because of Rasmus, which makes me very happy. So I'll give the word to Rasmus now, and then he can talk about his exciting work about monogenic and polygenic selection in humans. Thank you.
Thanks so much, Anders, for that introduction. Let me see if I can share my screen now. I think I can. All right. So you can all see that maximized, I hope. So, so one of the advantages of sort of becoming older is that you can allow yourself to give talks that are more sort of historical perspectives on things. So I'm going to do that today. I'm going to talk about methods and results of testing for natural selection in the human genome over the past 20 years. I'll focus mostly in the end of some recent research we've done in my group on polygenic selection, but I'll start talking more about where the field was 20 years ago to try to give some perspective of where we are today. So the first question when I ask is, well, why study human evolution, which is what we're doing here? And you can phrase that question in two ways. First, why study human evolution? Why is evolution in itself relevant and interesting? If you're a medical researcher or you're interested in functional aspects of the genome, why be interested in evolutionary biology? Well, there's a close connection between what is under natural selection and what is functional. We use that in bioinformatics all the time. We find regions of the genome that are highly conserved. We infer that because they're highly conserved, they're probably functionally important. That's really a statement about negative selection acting on the human genome, that these regions are important because new mutations are being weeded away by natural selection. Now, if we're interested in what is actually changing the genome or changing organisms, how organisms adapt, of course, that's not the type of selection we're most interested in. We're interested in positive selection that increases the probability that new mutation will go to fixation, that provides individuals that carry the mutation a fitness advantage. And much of what I will be talking about today are methods for finding those mutations. They're also very important for understanding functional relationships. Now, if you're in evolutionary biologist, you might ask, why study human evolution? After all, humans are rather awkward organisms to work on. We can't do experiments. We can't control anything. It's a varying environment. It's a pain to work on humans. We have a very long generation time. As a study organism, in many ways, not really great. We are probably as bad as it could be. Well, I would say there are two reasons why humans are actually a great model organism for other organisms. And one of them is that humans are spread all over the world, has adapted to many different environments. And that gives us an opportunity to study adaptation in many different environmental contexts. So that's one reason why humans are interesting. And the other one is a more practical one, is that there's so much functional information about humans, so much expression data, so much data about gene function that we can rely on. So when we study human evolution, it's very easy to connect evolutionary processes to function directly. And that's one of the reasons why I think many people that are interested in evolution have gravitated towards the human data, because the data was there and there were so many resources available. So the question is, how do we detect natural selection? If we're interested in understanding how organisms adapt, of course, natural selection is the key to that. And typically, we can't measure selection directly. We certainly can't do that in humans. It's very difficult to do in general, to figure out what are differences in fitness between different organisms. So what we're doing instead is we're looking at DNA sequences, and we infer the effect of selection indirectly by looking at the DNA sequences. And there are many, many, many methods for doing that, for taking DNA sequences and trying to infer where selection has been acting. And there's a very large body of literature on that. And I'm going to talk only about a few corners of this literature today. And you might not be surprised, that is mostly things that I've been involved in myself. But I want to make sure that everybody aware that there's really a very, very large literature of many other approaches to these problems than just the ones I'll be talking about today. I think the first sort of successful approaches that people started to use, where there really was a lot of papers coming out, where people were able to look at DNA sequences and infer natural selection, was by looking at DNDS ratios. So that effort started, well, almost 30 years ago, before you had genomes. The idea is that you take non-synonymous mutations, the ones that change the protein, and compare them to synonymous mutations, the mutations in protein coding regions that do not change the protein. The basic idea is that if no selection is acting, the rate at which the non-synonymous and the synonymous mutations occur should be the same. Negative selection against new mutations, would cause the DNDS ratio to be less than 1, and selection favoring new mutations, selections that provide a fitness advantage, positive selection, will cause the DNDS ratio to be larger than 1. And that kind of thinking provides a framework for comparing DNA sequences and asking which protein coding genes might have been under positive or negative selection. And I was so lucky that I was involved in one of the first large-scale studies of this Genome-Wide before we had many genomes, right after the sequencing of the human genome, before we had the chimpanzee genome. Private company Celera Genomics sequenced using Sanger sequencing what was then the annotation of all the protein coding genes in the chimpanzee genome, so we could compare to the human genome. It was through a collaboration with Andy Clark and Carlos Bustamante and other people. And so we're excited that we had now the opportunity for the first time to compare the protein coding sequences between humans and chimpanzees, and then ask the question, where has there been natural selection in these sequences. And of course, our hope was back then that, well, if you compare these sequences, we'll find all these really interesting genes where there's a clear signal of natural selection that will tell us about human adaptation, that will tell us about how humans evolved larger brain sizes, how we lost our body hair, how we evolved erect bipedal walk, and so on. That by comparing the sequences, we can this way elucidate important steps in human evolution. So we're very excited to do that. And when we did the comparison genome-wide of the NDS ratio, what we found was that the genes that showed the most evidence of positive selection, ones with the highest DNDS ratio, or at least the most statistical evidence of high DNDS ratio, tend to be genes that are evolved with immunity and defense in one way or another. There's some other categories too, but that was certainly a category that stood out. And that's something that's been replicated in many, many studies afterwards. And we think we kind of understand that there's this evolutionary arms race between pathogens and the host immune system, the genes in the host immune system. The pathogens evolve fast. The pathogenic environment is changing all the time. And for that reason, there's this very fast turnover of alleles in the human genes associated with immunity and defense. And you see this evidence of positive selection because of that. But what we didn't see, and what was very hard to find, was these particular individual genes associated with human-specific characteristics. That was something that turned out to be very difficult to find. And I think it's fair to say in many other studies subsequently on other organisms, people have been struggling with the same issue. And there are a couple of reasons for that. And one of the reasons is, of course, that there's statistical issues about power versus false positives and so on. But there's another more fundamental issue, that is that even if you find genes that have been evolving very fast, where the number we can show beyond any sort of statistical concerns that there is strong positive selection affecting, say, the human evolutionary lineage, it's then very hard to go in and figure out, okay, what is the functional effect of these mutations? And that's because if these mutations are fixed mutations between different species, they're not segregating in humans. We can't really do much to figure out what's the function of those mutations. What people have been doing is, of course, then go to model systems, trying to put those mutations into mice and so on. But it's, for example, if you're interested in human cognition, well, there's limits to how great a model system a mouse is for the way human cognition works. So there's a limit to this comparative approach of comparing different species. What we need really to be able to address these questions is we need population genetic data. And the reason why I need that is that now we have mutations that are segregating within a population. If we can infer which of those mutations under selection, then we can much more easily link that to function, because we have a whole host of different tools that we can use when first mutations are segregating to try to figure out what might their function be. For example, association mapping is one possibility. Which phenotypes are these mutations associated with? So that's sort of a general approach, I think, in the field, and one of the reasons why much of the research, I think, have focused after the genome sequencing have switched, at least my own group, switched a lot from comparing different species at the genomic level to really to focus on the population genetics. Because only at the population genetic do you have a good chance of really relating the selection that you see to particular functional effects, and really understand what is the effect of these mutations in a functional and perhaps even ecological context, which is what we're really after. Now, the problem with population genetic data is that demography is a big nuisance. So if we look at particular patterns in the data that might be caused by natural selection, very often the same patterns can be created by other processes of changes in population size, migration between populations. And you know, in real population, the pattern of changes in population size and geographic population structure can be very complex. So it can be very difficult to protect yourself fully against those kind of effects. If you see a particular mutation with an odd pattern, or a pattern that seems more compatible with selection than with some demographic history, how can we really separate the two? And I would say that's a problem we're actually still struggling with in the field. One thing that really helped us a lot is just simply the availability of whole genome data. And that's because we can focus more on outliers, the ones that show the most evidence of natural selection. The logic would be that if demographic processes, such as changes in population size, for example, if they're changing allele frequencies, that should affect the entire genome. So if you see just in one particular point in the genome, a pattern that's very different from what you see in the rest of the genome and is compatible with selection, we might have more belief in that actually being caused by selection now that we can compare to the rest of the genome than if we didn't have the rest of the genome. So genomic data has helped us to a certain degree. But we also want to do other things, try to maybe model the demographic processes to try to get at methods that are more robust. So the method we've been using in some recent study is a method that is implemented in this computer package Ohana. And basically what that package does is very similar to what is done in other methods available for identifying population structure, in that you can estimate the proportion of an individuals that belongs to a certain ancestry component. And most of you are probably familiar with this. So there's an example here in this slide. You have some different ancestry components, and then you have the proportion of those ancestry components in different parts of the world. And each individual in the top of the slide here represent a column, and the colors represent the proportion of that individual's, the fraction of that individual's genetic ancestry that comes from each of these ancestry components. So that's a basic model that is used in population genetics, a very generic model used in population genetics that can describe genetic variation among individuals, and at the same time allocate genetic differences between different groups that have been sampled. And what we're doing then is that based on this, jointly with this, we estimate a tree of these ancestry components. So we can now look at the evolutionary relationship between these ancestry components. And we can then use that as a background model where we model all the genomic variation in a population genetic sample using this, this tree, where we allocate each individual fractionally to each of these leaf nodes in that tree. That gives us a background model for describing genetic variation. And what we can then do is we can ask, are there any particular points in the genome where we find evidence that this model is not a good model? And particularly, it's not a good model because one of the lineages would have to be much longer to really fit the data. So for example, here, if we are interested in selection in the blue population, selection might have the effect of making the branch length longer. The branch lengths here, they are proportional to the amount of genetic drift or change in allele frequency. And if suddenly there's been selection in a particular locus in the genome, the branch lengths would be much longer because there's been a stronger change in allele frequency. So now we can establish a likelihood ratio where we can compare models that genome-wide model, the model that we have estimated for the entire genome, to a model where we allow that particular branch to be larger. And then we can very fast, using a model like this, scan the genome for particular regions or positions in the genome where the model with selection fits better than the model that doesn't have selection. So this gives us a very interpretable model for detecting outliers, that we're trying to find positions in the genome where particular populations or ancestry components have experienced a strong change in allele frequency. And we think those are good candidates for being loci that have been under selection. All right, so I'm going to give a little bit of the technical details about how we're doing that. So the way we model changes in allele frequency is using a Gaussian approximation. So that assumes that change in allele frequency can be approximated using a Brownian motion process. That's not really a great approximation if allele frequency are small or you're looking over very long time spans. But if allele frequencies are not too small, and if you're looking over relatively short time spans, this Brownian motion approximation is quite accurate under those conditions. And the advantage of that is then that the likelihood function, when you look at a particular locus, is given by a multivariate Gaussian. So you can describe the tree as simply a covariance matrix, where, for example, the diagonal elements, they correspond to length or proportional to length of these external branches. And if you want to test hypothesis of selection, that is an elongation of a certain branch, for example, the E branch here, you would do that then by multiplying that diagonal element corresponding to population E by a constant, so you get a longer branch associated with that ancestry component. So that's a little bit about the underlying machinery behind the method. But I wanted to talk about an application we did a couple of years ago, starting to become old, but I think it's such a cool example that I like to talk about it anyway. So it's research on these Bajau sea nomads that live in Indonesia. They also live other places in Malaysia, in the Philippines, used to also be in China. And it's a project that was started by a graduate student of Esquivilaslev, Melissa Hilado. And the project is really her idea. She came up with the idea of this. She really wanted to study these sea nomads and look at if there were any evidence of genetic adaptation to the very extreme and unique lifestyle that they have. Let me just explain that to you, what their lifestyle is. So they live as, they're often called sea nomads because they live on boats on the ocean in a marine, they have a marine lifestyle where they get almost all their food traditionally from diving, from free diving. We don't know how long time they've had this lifestyle, but there's some written evidence that at least 1,000 years, but perhaps much longer. They don't leave much archaeological evidence because they live on the ocean. So you don't find much archaeological evidence from them. So we know that they have had that lifestyle for at least 1,000 years, but we don't know, it could be considerably longer than that. So as they dive, people have, there's anecdotal evidence that can dive over 70 meters, and there have been anthropological studies that have argued that they spend 60% of their daily working time underwater. Okay, so what they do is they dive down, typically not for that long, actually, maybe half a minute or so, and then up again, and then down again, and then up again. But they keep doing that all day. So they expose themselves to extreme acute hypoxia. So hypoxia is deprivation of oxygen, again and again and again, for many, many hours, every single day. So that puts some really extreme demands on your physiology. And one of the things that Melissa was interested in was, okay, are there any biological adaptations to this lifestyle? And if you look at the genetics of the Bajau people, can we see any effects of that? So what to look at? Now, one of the issues with this kind of research project is that you work on indigenous people, and you can't really do anything that's invasive. You can basically, ask them things, then you can get samples of their saliva that you can do sequencing from, and then other things that you can sort of measure on the outside without using a needle, for example. But there's one thing you can look at that is not invasive, and that's certain issues associated with the diving reflex. And most of you might not be familiar with what the diving reflex is, but it's a reflex that all mammals have. And it's elicited by apnea, that's holding your breath, and cold water facial immersion. So you might be familiar with the feeling of, in the summer, jumping into the ocean or lake that's cold, and you get this feeling of being refreshed from the cold water. Now, that feeling is not just psychological. It's actually, there's a physiological response to this cold water immersion. And that physiological response includes bradycardia, which is a lowering of the heart rate, which lowers oxygen consumption. It also includes peripheral vasoconstriction, so it sends the blood into your central organs where it's needed most. And then there's also a contraction of the spleen. And why would the spleen contract? Well, the spleen serves as a reservoir of oxygenated red blood cells. So when the spleen contracts, red blood cells is being injected into the bloodstream, oxygenated red blood cells. And so it's a little bit like you having a diving tank with you. When you dive down under the water, you get more red blood cells carrying oxygen. So you can dive deeper for longer time with that. And that's something that we see in many mammals. And there are, for example, certain seals that have very, very large spleens. So the spleen of an elephant seal, for example, can weigh 100 kilograms easily. So they have massive spleens, and it's thought to be because of the adaptation to a diving lifestyle, where you also need to be on the surface. So when you're up on the surface, you can get all the red blood cells that are not needed. They can be stored in the spleen, but then when you're diving down, the spleen can contract and eject all the red blood cells. So one of the things Melissa was thinking about was perhaps in fact, these Bajau people could have had some selection associated with spleen size. So she lived down with her collaborators down in a Bajau village down in Indonesia for a little while, and worked with them on this project. And as part of that, she took buccal swabs to get DNA, and she also did these ultrasound scans, and that's what you can see her doing here, to try to measure the spleen size. This is an example of an ultrasound scan. You can't really see the spleen very easily. Here you can see the blue outline that's been identified. On the real scans, I'm told, I have never done this myself, but I'm told that it's easier to see the spleen because it sort of stands and jitters a little bit on these ultrasounds. But there's standard methods of measuring the size of the spleen because changes in spleen size is associated with certain pathologies for certain diseases. So there's standard ways of doing that using ultrasound. And Lisa took a course in this and learned how to do that, so she could do it down with her portable ultrasound machine down in Indonesia. And of course, the first thing when she came home she wanted to do was to compare was the spleen size larger in the Bajau than in other people. And so what other people to compare to? Well, she also took samples from some other people that were living in Indonesia as well, on the same island, Sulawesi, but living more inland, and had never had this kind of marine lifestyle. So these are the Saluans. And what you can see in this plot is that there's a lot of variation in spleen size among individuals. But in average, the Bajau spleen size is much larger than the Saluan spleen size. Of course, that doesn't tell you that the Bajau has adapted to have a larger spleen size. It could be that they have a larger spleen size for a host of other reasons, maybe because the diet that induces the spleen to become larger, or they've been exposed to certain pathogens that will induce the spleen to become larger, or something else. But there could be a genetic effect. It certainly opens up the possibility that that could be the case. So the next thing we did was we did a genome-wide scan, scanning what are the genes in the genomes of Bajau that seems to have evolved really fast, specifically in the Bajau, comparing them to the Saluans and to other populations, using the method I talked about before, based on these ancestral components and the phylogenetic tree relating them to each other. We took that lineage in the tree leading to the Bajau and we asked what are the genes on that lineage that are particularly elongated. And there was particularly one gene, or there was a list of 20 genes here that all had very high likelihood ratios, show evidence of selection. And there was one, when we looked at that gene, where the top SNP was also associated with spleen size. So there was an association there with spleen size as well. And so that suggests that the hypothesis is then that this gene would have been under natural selection, and by that having been affecting spleen size. I should say that if you notice the p-value here is 3 × 10⁻⁴. So marginally for one test, that's a small p-value. But of course, if you've done a genome-wide scan, this would not be significant at all. And I think this shows you the power of doing these selection scans, because when you're looking for selection, you find genetic variants that presumably are very important for example, physiology. And it gives you a candidate set of genetic variants that you can then test without having to do a multiple correction as if you've been testing the whole genome. Okay, so you can circumvent the multiple testing problem that reduces power in GWAS, in genome-wide association matching studies, so much by first identifying candidate genes that have been under natural selection. These other genes, most of these other genes, they don't show association with spleen size. They might be associated with other types of selection, perhaps associated with diving, perhaps associated with other processes in the Bajau. But this is a great candidate gene for being associated with adaptation to diving through an enhanced spleen size. So this just shows the evidence of selection. The top panel here shows along the length of the genome, the likelihood ratio. You can see there's this peak in a very specific region. If we look at association with spleen size, we see there's a peak the same place. The top SNP there is the same SNP, that's the top SNP for selection. And then if you look at expression data, and that's not for the Bajau, we didn't have that, but we have expression data for other populations, we can see that this particular region is a region that harbors expression variation for PDE10A, this gene here. so that the regulation of PDE10A is affected by genetic variation in this region. All right, so what is PDE10A? Well, it's involved in signal transduction related to alpha-genetic responses, like the contraction of smooth muscles, and the spleen contraction is a smooth muscle contraction. And then we looked at, okay, can we follow up on this and find something similar in other cohorts? This SNP, our top SNP, actually exists in other cohorts. And there was some marginal evidence also for an association in a European cohort that we found. So actually, you have done a GWAS in humans on absolutely everything, except on spleen size in healthy people. That doesn't seem to be something anybody ever would bother to measure. So what we had to do is make a collaboration with some people that had abdominal scan as part of their cohort. And then we could measure spleen size from those abdominal scans. And so we could do a bit of a follow-up that further lowered the p-value when we did that by looking at the European cohort. And what Melissa then has been doing for her postdoc, she's in Utah right now in a postdoc, she's been working on a mice system where, and I'm not going to steal away too much of her thunder by talking too much about yet unpublished results, but all I would say is that she's able to mimic the effect we see in the Bajau by modifying the expression of PDE10A, and that then also changes the spleen size in mice. So I think that's quite exciting, and I think it's good validation that in an experimental system like that, you can do that. That also shows against the weakness of working humans. Of course, you can't do validation experiments really in humans for real. You can try to do it maybe in cell lines, but if you're looking at something like spleen size, it doesn't really work. So you have to go to other model organisms. Okay, so we can look at also, we can try to model when might the selection has been acting, and using approximate Bayesian computation, we find that the divergence between these populations is about 15,000 years, we estimate, plus minus. And of course, this estimate relies on strong assumptions about demographic model, but it gives you an idea about the amount of divergence between them. Should also say that Salomons might not be the most closely related population to the Bajau. It's almost certainly it's not, but it's the most closely related that we used in our study. And from that, we can then also estimate some estimates of how strong the selection was, and our estimate of the selection coefficient was that it was 0.5%. So that's the difference in fitness between people who carry the mutation and people who do not carry the mutation. And then our estimate was that when selection started, the initial frequency of the mutation was about 2%. And that matches well with the fact that we see that this mutation also in other populations at very low frequencies, 1-2% typically. But it's much higher in the Bajau. All right, so that was the case of the Bajau. We think that selection there, through mediated, at least in part through this gene, has led to increased spleen size in the Bajau, presumably to give them better diving abilities because of the ability of the spleen to store oxygenated red blood cells. Okay, so that was the Bajau example, and it's sort of one example of many studies that our group and many other groups have been doing. We're trying to scan the genome and we try to find the genes that have been under selection, and you're trying to relate that to function and to understand the relationship between natural selection at the genetic level, what's going on at the organismal level as well. But of course, a limitation of many of these methods, certainly this method that I talked about, is that it uses relatively little of the information in the data. We're going SNP by SNP, and we are asking, are there difference in allele frequencies between population? Where's the most difference in allele frequencies? And it's relatively limited because it's only using this marginal allele frequency information from SNPs independently of each other. Now, if you really want to use more information, you have to think about more carefully about the process that generates variation in a genome. And the way in population genetics we model that is through these coalescence trees. So we can model the whole history of a sample through a coalescence tree. And back in the old days where we just did mitochondrial DNA and so on, this was an easy thing to do. We could do that very easily. But with genomic data, that becomes a problem because each position in the genome might have a unique tree because of recombination. So in contrast, when you're studying different species that are highly divergent from each other, you can, to a good approximation, assume that's just the same tree relating all the species to each other. But if you look at individuals within a species, every single position in the genome might have a unique tree. There are millions of different trees. So this led us to think that, okay, it's not really possible to fully get all of the information out from the genetic data using these coalescence approaches, these genealogical approaches where we look at the coalescence tree. It's not computationally feasible. So we tried many other approaches for essentially approximating a likelihood function. A likelihood function is a function that has all of the information in the data. So you often want to try to find that. But it wasn't really possible. It's technically to actually do that except by approximations. And there are large literature and various ways of trying to, for example, detect selection without explicitly using all the coalescence trees, without full likelihood methods, just because technically it's too difficult to do. So that's at least what I used to think until this paper was published and landed on my desk. So this is a paper from Adam Sibbel's group that developed a method called ArcWeaver, which shows that it is in fact possible to sample what's called ancestral recombination graphs. So that's the collection of all these trees throughout the entire genome and the points in which there's recombination between the points where there's recombination that generates these trees. It is possible, in fact, to sample that genome-wide for a sample of reasonable size, for example, 100 individuals. It's not easy, but it's doable. It is possible using a method that they develop in this paper. And with that opens up then the possibility that you can do full likelihood methods, because when first you can sample these ancestral recombination graphs, these sets of trees from the entire genome under one model, then you can use various statistical tricks to then get the likelihood function on the other model, something called importance sampling. So we started working on that, and that led to this paper by Aaron Stern. And most of what I'm going to end up talking about here, the end of the talk, is work within the last couple of years by Aaron Stern of using that kind of thinking. So the basic idea here is that you want to first sample all these ancestral recombination graphs. So these are collections of trees. Okay, so we can think of that as the collection of trees as a latent variable, a variable that we don't know what values it takes. And it's very, very complex. It's a collection of all of the trees for the entire genome. And we want to integrate all of that. That is, in theory, we wanted to take every single possible trees throughout the entire genome, calculate a certain function over all of these trees, and then summing over that, integrating all of that. That's not possible, but we can sample. And methods such as the one published in ArcWeaver will allow us to do that sampling. Then the second hidden layer, that is, we then need to model for selection how the allele frequency changes through time. That we can model using models similar to hidden marker models, where at different points in time back in the history of the sample, we model what the allele frequency is. It's another latent variable, another unobserved variable that we also integrate over. That's really easy to do because we can use the type of dynamic programming algorithms that will allow us to do that very fast. So when first we can sample these trees, when first we can sample these ancestral recombination graphs, this collection of trees in the genome, then we can in fact do full likelihood estimation. That is, we can make statistical methods that use all of the information in the data to get at where there might be selection. So Aaron implemented this in a heroic effort, and then this is sort of some figures that illustrates the results of it. So you get a likelihood function out. So this is, for example, a likelihood function here in figure D, the top slide here, that shows a likelihood function when there's no selection. So there's a peak very close to 0, and the true selection coefficient with the 0 is not very different from the one that's been inferred, and there's no real evidence of selection. The evidence is in the log likelihood ratio, which we also calculate. Below it is an example where there is selection. Again, we can estimate the selection coefficient reasonably precise. That is, remember, the selection coefficient is this difference in fitness of people who carry the mutation versus people who do not carry the mutation. We get a nice likelihood function here, and we can do maximum likelihood estimation of the selection coefficient. We can also then, because we model explicitly allele frequency back in time, do a kind of posterior decoding then, where we get this path of the allele frequency. So the top one here is the path of the allele frequency for the neutral case, where there's not much change in allele frequency going backwards in time. When you go to the right here, that's backwards in time. But you can see in the lower case here, there's strong change in the allele frequency compatible with the fact that there's been selection acting on the mutation. The red lines here, that's the true allele frequency, and the coloration in yellow, the arrows in yellow is where we infer this high probability that that was the allele frequency back in time. So we can infer the selection coefficient, and we can infer the trajectory of the allele back in time with this method. And we can ask, okay, how much power does this give us to identify selection? So it gives us a tremendous amount of power over similar methods. So these are all methods that just use one population. You're not trying to compare to other populations like what we did in Bajau. We're just looking at one population, ask from just the data in one population, how much evidence is there for selection? And in the red line, we have Aaron's method clues. And then in the blue lines is another method called NSL that uses haplotypes. And it's very similar to another method that you might be more familiar with that's called ISS. They're very similar to each other. The NSL should have a little bit more power under certain scenarios than ISS, but they're otherwise very similar. And then in yellow, we have ChatGMSD, which is a very classical method for trying to use the same kind of information to detect selection. It's a method we always like to include because it doesn't have very much power. So whatever method you develop always looks good in comparison to that method. So we also included it here. So these are curves that show the true positive rate, so the rate at which you actually find selection on the x-axis as a function of the false positive rate, on the y-axis as a function of the false positive rate on the x-axis. So good methods should go all the way up to the corner of 0.1 in a figure like this. And what you can see is that on all the scenarios we have looked at of different selection coefficients and different allele frequencies, there's much more power when you use the likelihood function. That other methods that use less information in the data, they also have much less power. And then in fact, certain scenarios, if the allele frequency is relatively high, but the selection coefficient is small, where you have a tremendous amount of power using likelihood function, and existing other previous methods really can't find selection at all. When you follow the diagonal here, that means that you have essentially zero power to detect selection. So we're quite happy with that. The method seems to be quite powerful. It relies on assumptions about demography and so on, and we're not making strong claims about how robust it is, although it doesn't seem to be less robust than other methods that we have compared to. There's an application of it. We looked at the lactase gene. You might be familiar with this story of selection in lactase, presumably as an adaptation to dairy farming. If you have a certain allele in the lactase locus, you're able to digest milk as an adult. And if you don't have that allele, you're not able to, and you have lactose intolerance. And there's supposed to have been strong selection for this allele in Northern European populations, but also some other populations, other places in the world. And what this slide here shows you is our inference of the allele frequency in this lactase locus back in time, just from the modern data. And that's the black line. And then there are also some dots here. Those dots represent European data where people have either seen or not seen the allele. And then in red, you see the inference in a paper by Ian Matheson, where he tried to analyze using this ancient DNA, what was the allele frequency back in time. And you can see that his inferences and our inferences, which is just from the modern data, are very similar to each other. We estimate that back in time, the allele frequency was a bit larger, about 5,000 years ago, than he estimates. And it's possible that the discrepancy could have something to do with population structure. That, in fact, the ancestry of the salmon we look at right now today might not be the exact descendants of these individuals 5,000 years ago. That's in fact unlikely. So this could have something to do with it. Okay, so yeah, so these results are from Madison 2018. So we can also look at other loci here, a bunch of loci that's associated with pigmentation. It's also previously been analyzed, and we can calculate this log likelihood ratio that provides evidence of selection, the selection coefficient, and also the allele frequency pattern time. And these are all genes that have been claimed to be all under selection. You can see some of them we find no evidence of selection, like TTC3 here, for example, selection coefficient of 0, log likelihood ratio of 0, and no change in allele frequency back in time. That doesn't seem to have been selection on that locus, for example. There's another one. There's one of several candidate genes in the TYRP1 gene that's been proposed to be under selection. We find no evidence of selection in this one. Again, our estimate of selection coefficient is 0, log likelihood ratio is 0. And in fact, this analysis is all done for a European population. But if you look around the world, you find that this allele has a relatively high frequency other places too, Africa and other places. So again, that's compatible with, well, at least there haven't been strong localized selection in this gene. The allele is old and has had a relatively high frequency many places in the world for a long time. Doesn't seem to have evidence for a lot of strong recent selection. Here's some genes or some alleles that seem to have evidence for selection recently. ASEP, KRGLG, TYR, they all have high likelihood ratios, strong evidence of selection the way we measure it. And if you look at the geographic distribution, you tend to see these alleles in Europe. And then also you see them a bit in the Americas of people in the Americas that trace some of their ancestry also to Europeans. So these seem to be alleles that only have high allele frequency in Europeans or people that are descendant from Europeans. Okay, so that's a bit of methods for detecting selection. And we're quite excited about this new method using ancestral recombination graph. But you could say that the whole sort of project what we're doing here of going in and finding individual mutations under selection is a bit challenged by the fact that we know that most phenotypes we're interested in are really highly complex and are affected by many, many different genes, many different genetic variants. So it raises the question, does adaptation proceed through fixation of alleles of strong effects? Which is implicit we assume when we sort of scan the genomes for where in the genome do we find some genes that have been under strong selection. And I think the field of, at least of human genetics, is moving more and more towards a consensus that that's probably only rarely the case. That we think that based on what we know of complex genetics in humans, that's most likely adaptation probably mostly proceeds in a polygenic fashion. That is, by many mutations of weak effects that affect a trait. So a polygenic trait is a trait where multiple loci affects the same trait. And most of the traits that we're interested in are really polygenic traits. There are very few that are monogenic, that are affected by just a single gene, or there's just a single allele that might affect it. So this whole viewpoint of, okay, we want to find the mutation under selection doesn't really, is really not very useful, I would say, perhaps, for much of the adaptation we're interested in. So polygenic traits, we need to deal with them. And there are more complications. It can be epistasis. These different mutations in different loci, they may interact. This interaction of different mutations is called epistasis. Then there are probably multiple traits. I mean, so depending on how we measure things, there are typically multiple traits that somehow interact with each other. And typically, one mutation might not just affect one trait. It will typically affect multiple different traits. That's what we call pleiotropy. And the way we measure phenotypes in humans or other organisms, most genetic variants that have an effect on one phenotype typically also affect other phenotypes, the way we measure it. So basically, everything we're interested in is polygenic and pleiotropic. And very little of what we're doing acknowledges that. Although there has been a push lately in the field to really look at polygenic selection. But it's complicated and it's difficult, and there's been challenges in getting at polygenic selection. Now, one of the advantages of the approach I talked about before that Aaron Stern has developed is that when first you have a full likelihood function, it becomes much easier to also model other, expanding the model, for example, also to do polygenic traits. And so we've been recently been working a lot on that. And the basic idea in how you model polygenic selection is that if there is selection favoring a trait to change, it will very fast move to a new option or optimal. But there's be this transient phase where selection works as positive selection towards the new optimum. And very old research back from the 70s shows that the selection coefficient here on the left affecting a single locus is approximately equal to the selection gradient omega affecting the entire trait times the effect size of the mutation. If you measure if the effect size is standardized relative to the total variation in the trait. So that also, in some sense, some of all the effect sizes will be related to the heritability. The heritability is also in here. So through that equation, we can now write a likelihood function, not for the selection coefficient, but for the selection gradient acting on the trait, the amount of selection acting on the trait, by simply taking the product of the likelihood function for all these individual SNPs, expressed here as a function of the effect sizes that we need estimates of. So this is something you can do when you have, for example, done a GWAS and you have estimates of the effect sizes. And then you want to ask the question, has there been selection acting on particular phenotypes? By looking at what's going on in all of the genetic variants that affect that phenotype. Okay, so Aaron Stearns implemented that method, and when I say he implemented that method, there's a lot of things under the hood that I don't really have time to talk about, not so easy to do, but he managed to get a method working in the end. And I'm just going to start showing you some plots from simulations of the performance of that method. And what we're comparing it to is this method here out of Jonathan Prince's group, the SDS methods, or on traits, the TSDS method, which we compare to because it's state of the art. And it's a very, very clever method that was developed. We're going to sort of compare our method, show it's better than that method, not because this method is bad in any way, but just, I think, by the power of having a full likelihood function, what that gives you over other methods that can't use as much of the information. So for example, here is a power plot to the left, and the blue method is this previous state of the art method, the TSDS method. On the x-axis is the selection gradient, on the y-axis the power, so the probability that you will actually detect selection. The blue one is the previous method, that's our methods, which we call PORM, polygenic adaptation likelihood method, is in the red. And you can see there's an extremely strong increase in power here. This is when we simulate first a GWAS with 100,000 individuals in the GWAS. We mimic how SNPs have been identified in the GWAS, so we have all the biases of ascertainment and so on associated with the GWAS incorporated into these simulations. We have a heritability of 45%, and then the genetic variation is distributed under 1,000 loci equally. So there is highly polygenic with 1,000 loci contributing to the trait. And then the sample size we have from the PORM method, this is because we're using 1,000 genomes data there on 178 individuals is the sample size we use there. For the TSDS, we used 6,390 individuals that was used on some UK Biobank data that's been analyzed previously using the TSDS method. So a much larger sample size for the TSDS method. It can deal with a much larger sample size than we can, but there doesn't seem to be much of an increase in power from that. We also get from our methods maximum likelihood estimates of the selection gradient acting on the trait, and that's what you see here. The dotted line, that's the true values, and our estimates is in the red, in red here, and it seems to perform reasonably well at getting decent estimates of what is the selection gradient of the trait under these conditions. Now, one of the major issues when detecting polygenic selection is that biases in the estimation of the effect sizes from the GWAS can strongly bias your inference of selection. Particularly, the classical problem in GWAS studies of population structure will also affect you here. So there have been several previous studies on selection in humans that have been challenged by this. For example, there was many papers that first claimed that there's been selection for increased height in Northern Europeans and perhaps decreased height in Southern Europeans. Now, that was later shown possibly to be due to a stratification effect where the GWAS samples has a mixed ancestry of people from Northern and Southern Europe. And they're also having to have maybe different diet, different living conditions. So the difference in height, not because of genetic factors. And this creates this bias in the effect size estimates that also will affect selection studies. So what we're doing here is we're simulating this effect. We're making a mixed sample of individuals from Northern and Southern European ancestry. And then we vary how much of the total variation that's caused by uncorrected genetic stratification, that is differences between these groups. And again here you have our method in blue and the trait SDS method in red here. And I would say both methods are actually quite robust to this. So this is in terms of standard deviations of how much of the total variance that's explained by the stratification effect. So you need quite a lot of uncorrected stratification before these methods start to give you false positives. I should say, sorry, the red one is our method. The blue one is TSDS. I think I got that wrong. The red one is PORM. the method developed by Aaron. So both methods, I would say, are relatively robust. You had to have really strong uncorrected stratification, at least the way we've been analyzing it, before you really start to getting a lot of false positives, and we seem to be somewhat more robust than the previous method. Another thing to look at is what p-value threshold you use for the mutations that you include. The reason why that's important is that many of the biases come in from choosing the top SNPs if you choose too many SNPs, because then you get all these SNPs that haven't been under selection and might not affect the trait, and where the effect size is poorly estimated. And then the way you choose your SNPs and all these biases becomes really, really important. So as the p-value threshold for which SNPs from the T1s to include in your study lowered, we can see that TSDS start to get a lot of false positives, but we are relatively robust to that. We can go back down to 5 times 10 to the minus 4 as a cutoff, include a lot of SNPs, and still not have that many false positives be above the nominal level. We, for all of these, have a little bit more than the nominal 5% false positive rate, but only very little increase in false positive rates. Okay, so I'm going to talk about some real data examples, and for that, and actually for everything I've been talking about so far, the way we are getting the genealogies, the trees, is actually using this method developed by Leo Spidel in Simon Meyer's group. And the reason why we're using that method instead of ArcWeaver is that there were some biases we find in ArcWeaver that this method doesn't seem to have, and also it's much faster. So it helps us a lot. It's very slow to sample these genealogies. It's much faster. Now, this method is not fully sampling all stochastically the topologies in the way that ArcWeaver does. It's less ambitious. It just estimates some trees and then it samples coalescence times. But for our purpose, that actually seems to perform quite well nonetheless, just to do that. So the approximation that we have in this method actually seems right now to be what works the best for us at the moment. Certainly much faster than for example, ArcWeaver that we previously used. So we can look a little bit what's the effect of actually not knowing what the true tree is, but in this case, sampling it from these relate trees. So this method here is called relate, for this method for sampling coalescence trees. And what we are comparing here is two simulations, one with a selection, one where there's no selection. And then we assume first in pink, what if you had the true trees, you knew them, and you knew which steps were causal. So you didn't have to select the causal steps, that was known. And then the pink here is what we get in neutral case, and here when there's selections. The method works very well. Then we can ask, what if we estimate the relate trees instead? So we don't know what the true trees are, we estimate them using relate. And the method still performs well. There's some reduction in power from that. We lose some information from that. There's not as much information as in the estimated trees than there actually were in the true trees. That's not surprising. Also, the coalescence times are being sampled. We don't actually know the true coalescence time. We're estimating a posterior distribution of those that also leads to a reduction in power. But still, we perform pretty well. And then what if we don't know what the true trees are, but instead we have these tag SNPs that have been identified using GWAS? That performs pretty much just as well as if we if we knew the true SNPs. So there doesn't seem to be much of an effect on the method and reduced power from the fact that we have identified SNPs in the GWAS rather than actually knowing which ones are true. And I should say in all the simulations I showed before, we're actually modeling this fact anyway, that there's uncertainty about what are the true SNPs, and in many of our simulations, we've sampled SNPs that are tag SNPs that are actually not the true causal SNPs. All right, so what happens? So this in real data, so we chose a set of phenotype from UK Biobank and analyzed that to figure out where do we find evidence of positive and negative selection. And I'm just gonna, this is hard to see here, so I'm gonna zoom a little bit in on the tails to look at the traits that we see there. So some of these traits have been found before to be under selection, other ones not. So the ones to the left are the ones where the selection against the trait, the way it's defined, and the ones to the right is where the trait has been enhanced. So the phenotypes we find are things like sunburn, hair, tanning. So these are all associated with pigmentation. We also find age at first birth, been found in previous studies, educational attainment, how much education you have received, for example. We also get HbA1c, which is glycosylated hemoglobin. That's associated with type 2 diabetes. And we get a behavioral trait, neuroticism, as the traits where we find evidence of selection. And some of these results are new, some of them old. The pigmentation one, all the people have found, educational years, certainly, age at first birth of traits that people previously have been pointing to. Now, one advantage of this method is then that also that you can extend this to model pleiotropy, this interaction or this fact that a simulation might act on multiple different phenotypes. So you can start asking the question, okay, if we saw selection acting in one trait, is it really selection that favors that specific trait? For example, glycosylated hemoglobin is associated with all kind of adverse effects. Okay, so is it working directly on that, or is it working on something else? Educational attainment, has selection really been acting on increasing people's education? That's what we're measuring. But we can then measure many other traits and see, okay, what does selection seem to have been acting on? And the way we can do that is that we can make a similar likelihood function to the ones before, but where we sum over traits. Okay, so we can sum over traits or write a likelihood function similar to the one we wrote before, where the likelihood acting now includes this selection affecting or effect sizes for each of all of the different loci and all of the different traits. So you can, using classical quantitative genetics, make a combined model for multiple traits. So what's the effect of that? So to look at what happens if traits are genetically correlated, what happens? And so we simulated trait selection on a trait we call trait 1 here. And we look at 3 other traits, traits 2, 3, and 4. And this here is how much each of these traits is genetically correlated with trait 1. So genetic correlation between trait 2 and trait 1 is 80% here. And the genetic correlation between trait 3 and trait 1 is 50%, and so on. And now we can see what would happen if we ignore that and then test each of these traits separately. Well, the strong selection on trait 1, we have 100% power essentially to detect selection on trait 1. But trait 2, which experienced no selection itself, but was correlated with trait 1 genetically, we also find selection on that in 80% of the cases. Okay. And similarly for trait 3, we find evidence of selection in more than 40% of the cases. If we analyze the traits one by one, okay, so that illustrates this concept that selection acting on one trait could really be selection on acting on another trait, but then there are correlations. Okay, but if we know all the different traits, we can then correct for that. So we use this model that Aaron developed in a joint test where we analyze all the traits simultaneously. We will have strong power to still detect selection on trait 1, but very little power or we don't get any false positives for selection on the other traits. Now you can see that's a reduction in power. That simply comes with the extra parameters, the fact that some of the effects we see can be allocated statistically to other traits. We have many more covariance, it leads to some reduction in power. So we have some reduction in power, but we don't get these false positives on the other traits. So we applied that also to some data. And I'm just going to show you, or to the same data, so some of the examples of traits that really changed and changed strongly in terms of how much selection we infer. So for example, we have here type 2 diabetes. So type 2 diabetes, if we test it marginally, it has no evidence of selection. But if we condition on glycosylated hemoglobin, on HbA1c, then there's actually a much larger z-value and a significant p-value. And this test over here, the 5th and 6th column, is a test that tests whether there's a significant difference between the z-values of the two tests, whether we do it unconditionally or unconditionally. We see there's a significant difference. We suddenly now have much more evidence of selection on type 2 diabetes. So that's interesting. There's lots of hypotheses about selection on type 2 diabetes. It goes back to this paper back from the 60s by Neil. He proposed this 50 genotype hypothesis, where he hypothesized that things like type 2 diabetes, traits like that might be a high frequency in some populations because in a previous environment, many of the phenotypes associated with that disease, they were advantageous. For example, metabolism, so that you would use the energy resource more efficiently and you would be more storage up of energy and so on. So things that today might be associated with, for example, obesity, might have been in a previous environment where there were less resources available, there might have been positive selection for that. So if we go back to our results, HB1A1C, the glycosylated hemoglobin, is strongly associated with many of the negative fitness consequences of type 2 diabetes, of many of the effects on the cardiovascular system. So one interpretation of this, and this is now pure speculation, but one interpretation is that some of the effect of type 2 diabetes that have to do with more efficient use of energy, other things like that, that now increases the chance of type 2 diabetes. That's actually in the past been under positive selection. But then there's been negative selection, strong negative selection, then acting on the immediate negative fitness consequences of genes associated with that, immediate negative fitness consequences of diabetes-related phenotypes. So that's one possibility. Another results that we see here is, we have two other results here where things change quite a lot. Hair color, which is this hair, when you condition on tanning, that is a odd phenotype that is associated with skin pigmentation, of course. That didn't seem to show any evidence or much less evidence of selection when conditioning on tanning. So that might be, that's an issue. The hair pigmentation, skin pigmentation has been extensively discussed. Why is there selection on all these pigmentation-related genes? And there are basically two types of hypotheses for this. One is it has to do with vitamin D, that if your vitamin D is being produced when you get exposed to UV light, but if you get too much UV light, that can be damaging for you because you get sunburn and might cause cancer and so on. So if you're in a climate where there's not much UV radiation because the sun is not shining so much, we're living in Ireland or somewhere in the British Isles and the sun is not shining most of the time, then it's advantageous to have lighter skin color so you can produce more vitamin D, and there's not such a big fitness consequence of the increased risk of sunburn and skin cancer. So that's one hypothesis. That probably is the main hypothesis, but there's also a lot of suggestions in the literature that it has to do with sexual selection, that somehow make preferences for individuals with certain types of pigmentation that they have been preferred. So our results show that if you condition on tanning, which is this skin pigmentation related to response to exposure to UV light, if you condition on that, you actually don't find selection for hair color. Then in fact, that would suggest that the change in hair color is predominantly a correlated response with the selection associated with tanning. So that would suggest it's not sexual selection. It is in fact selection associated with this exposure to the damaging effect of UV radiation versus vitamin D production. Now, last trait that we looked at, where we also found some interesting results, was looking at educational years. So amount of education. So several different studies that have shown that argued that there might be selection associated with increased educational attainment. What we found, the odd result that we found, that when you take educational attainment and then condition on certain traits, particularly, for example, sunburn, it's another slightly odd trait to have here. But if you condition on that, much of the evidence for selection on educational attainment goes away. So sunburn, that is a phenotype where you ask people, how much have you experienced sunburn? How likely are you sunburn? You just ask people of that in a questionnaire. When you condition on that, that doesn't seem too much selection on educational attainment. Now, why that is, again, is speculation. But one hypothesis that you could postulate is there's such common genetic mechanism that has to do with whether you get sunburned or somehow mechanistically and some cognitive properties. That's probably highly unlikely. But there's a strong correlation, genetic correlation between educational years and sunburn. It's likely mediated through various environmental effects. One possibility, of course, is that it has to do with how you've been treated in society, depending on your ability or depending on your skin pigmentation. That's one possibility, that you are disadvantaged in terms of educational achievement, that relates to your skin color. So that's a possible explanation here for these results. But I should also mentioned there could be other explanations for this, and we don't really know. This is all speculation. But the fact is that the educational selection on educational attainment, when you condition on some of these skin pigmentation related phenotypes, particularly sunburn, much of that result seems to go away. So I end with that. I will make sure to remember to thank all the people that have been working on this project, in particular Jade Cheng here first, who worked on developing Ohana, the first method I talked about, and of course Aaron Stern, who has done all this work on the maximum likelihood methods for finding selection using ancestral recombination graph, both the monogenic and the polygenic work, and Melissa Elado, who was the one doing the work with Lebajau on selection for increased spleen size, and many other collaborators, including several people in Copenhagen who worked together with this, Ida Malke, Thorfinn Corneliusen, Rasmus, and other people. And thank you to all of you for listening to me today. That's it. I guess questions now. I don't know quite how this works.
Okay, so I just got unmuted. So first of all, lots of claps. I'll be the only one you can hear, but I'm sure people are
well, thank you so much, Anders.
So what we'll do now is you can raise your hand inside the Zoom. So if you go to participants, then you should be able to press a raise hand button. And once you do that, then we'll unmute you. So I see there's already one question there. And then, so Fernando will do that. So once you get unmuted by Fernando, then feel free to ask a question. And
yes, thank you, Rasmus. Can you hear me?
Yes.
Good. So you have shown that it's possible to model pleiotropy, but is it possible at all to model epistasis?
And that's a good question. So in this framework, we're not doing it right now, but I think in fact it is. You could do that. We, and I'm sure many other people are working right now on trying to figure out how much epistasis there really is in some of these, from these human data. We have these massive data sets on that right now. We can do lots of studies on epistasis. Now, epistasis is difficult. Epistasis is many different things. There's sort of pairwise epistasis, which is notoriously difficult, or three-way epistasis, notoriously difficult to find because it's like a needle in a haystack. So you have all these pairwise comparisons. So I think it'll be very hard. You could do that here, but really the statistical evidence when you correct for multiple testing of the pairwise epistasis is typically very low. And I think just because you have this massive multiple testing problem, we have a hard time often finding the primary effects, and then these interaction effects where there's so many multiple pairwise comparisons is hard. But I think the kind of model that I think that maybe you will get to the more what are called global epistasis, where there's sort of, you think there's a positive or negative synergistic effect between many mutations. So that right now most models, including this model that we're using here, assumes that the response is additive. So that as you have more mutations, all the effect of all the mutations just add up. But you could have different functions that relates that. So you could have different epistasis but that function that relates the response of different individuals is not linear. And I think that kind of that's also a type of epistasis. And I think that kind is more computationally attractive to model. And the question is, how important will it be to do that? For some traits, it might be very important. For other traits, not. For example, we do some studies, it looks at something like height. That does seem to be very, very additive in its effects. It's very hard to reject an additive model for example, human height. But other traits it might be a different story.
Thank you.
Thank you, Rasmus. I have a question from the chat here, from Marta here in Copenhagen. It's a technical question. It says, what's the minimum coverage we need to run this kind of analysis? And I guess if you can talk anything about the technical aspects, if we need phased genomes, haplotypes, or coverage issues.
Okay, so that's a very good question. So I didn't talk about that. For the first kind of method, you can have very low coverage. We incorporated some ideas actually from Anders Albrechtsen, that you can use genotype likelihoods when you do these admixture models. So there you can do very low coverage. Of course, if you have too low coverage, sometimes various forms of validation of the data becomes harder. But in principle, you can go very low in the coverage. For the methods I talked about in the end, the ones developed by Aaron Stern, these full likelihood methods, right now, the tree inference methods assume that you have relatively high quality genomes that can be phased. And so that's a major limitation. The question is how much, I mean, the limit here is our ability to estimate distributions of coalescence trees. And right now, we can only do that really well for high quality data. But I'm sure that in the future, we'll be better doing it for low coverage data. So for I would say, if you want to do the kind of thing I did here for the human data, you know, you need a coverage, I would say like 15x or something like that to really be able to do that well.
So if there's no more questions from the chat, then I'll ask you a question. So so there's lots of ways to improve our way to detect selection. So we could have more individuals, we could have deeper sequencing of more individuals, we could have better phenotypes, we could have better sampling of more diverse people, or we could have ancient DNA. So what do you think is the limiting factor right now, and where should we spend our time doing better?
Oh, that's a good question. And I don't really know what the answer is to that question. I mean, I can tell you what I'm excited about. I'm excited about that you have ancient DNA now. So of course, what we're doing here is we're explicitly modeling as part of it. We're integrating out this path of allele frequency back in time, and we can now also incorporate ancient DNA in that. It's trivial to do that. So you also have ancient DNA in this path back in time. And I think it looks like that really does give you a lot more power. And so I think all these ancient DNA analysis will help a lot. I think sample size in terms of number of individual for detect selection is probably not the problem. I think often we're limited by what you know more about than I do is all the estimates of effect sizes in these GWAS. That is, you know, very limited. You know, many of the traits we're interested in, they have all these very, very small effect sizes, highly polygenic. And, you know, if we could get better estimates of effect size, that would probably increase the power for the polygenic selection a lot. But we're already looking at 500,000 people or more for the effect size estimates. So there'll probably be a limit to that. You know, we were taking everybody that lives on Earth and measured them very carefully. There's still a limit to how well we know the effect size of all these mutations are very weak effects. Although having 1 billion people is certainly better than 500,000.
Yeah, that would be great. Is there any other last questions? I don't see any raised hands. Then
well, thanks so much for having me.
Yeah, I found
bye-bye, everybody.
I'm clapping now. Yeah, so thank you very much, Rasmus. And on the 19th, we'll have the next talk with Benjamin Peter. So hope you tune in there, and hopefully we'll do better with the technical issues. And so I want to say thanks to a lot of people who tried to fix some of the technical issues. And unfortunately, we didn't fix all of them, but we'll do better next time. And with that, I think we'll stop now. So thank you very much for coming, all of you. And hopefully see you on the 19th.